is Shwetha, and I'm going to go over the essentials of DNA structure and the steps involved of DNA replication. To begin, a nucleotide is the main building block of the nucleic acids DNA and RNA. In DNA, the nucleotide is composed of a deoxyribose 5-carbon sugar, which is this, as well as a phosphate group and a nitrogenous base. The phosphate group consists of one atom of phosphorus surrounded by 4 atoms of oxygen. The nitrogenous base is located on the other side of the sugar. In DNA, these bases can be adenine, thymine, cytosine, or guanine, abbreviated A, T, C, and G respectively. In RNA, however, uracil is present while thymine is not. Furthermore, these bases can either be categorized as purines or pyrimidines. The polymer DNA is a type of nucleic acid made up of a bunch of nucleotides linked in a chain through phosphodiester bonds, which is between the 5-carbon phosphate and 3-carbon of the sugar, making a polynucleotide, which then makes DNA. Nucleotides are the building blocks of all nucleic acid. Each nucleic acid has a 5' end and a 3' end. This refers to the 5' carbon and the 3 carbon on the sugar. You can tell if it is a 5' end because it will have a phosphate group attached, and the 3 carbon has a hydroxyl group. The two strands of DNA are always arranged anti-parallel to one another. So if one strand goes from 5 to 3, then the other strand will go from 3 to 5. The first step in DNA replication is to unzip the double helix structure of the DNA molecule. This is carried out by an enzyme called helicase, which breaks the hydrogen bonds holding the complementary bases of DNA together, A with T, C with G. The separation of the two single strands of DNA creates a Y shape called a replication fork. The two separated strands will act as templates for making the new strands of DNA. Single-stranded binding proteins is attached to these post-replication fork single strands of DNA, preventing them from reforming the double-stranded state. The new strand made over here will be called the leading strand. The enzyme primase makes a short piece of RNA called a primer, which then comes along and binds to the end of the leading strand. RNA primase can attract RNA nucleotides due to the hydrogen bonds between the bases, adding them one at a time to the nucleotides to the 3' end. The primer acts as the starting point for DNA synthesis of the 3' to 5' parent chain. DNA polymerase III then binds to the leading strand and then walks along and adding new complementary nucleotide bases A, C, G, and T one at a time to the strand of DNA in the 5' to 3' direction all the way down the nucleotide. DNA polymerase I then reads the fragments and replaces all the RNA primers with complementary DNA nucleotides. The lagging strand is synthesized away from the replication fork and is done so discontinuously. The lagging strand happens at the same rate as the leading strand, but its synthesis is slightly delayed because each new fragment of this strand cannot be started until enough template has been exposed at the replication fork. To begin, the enzyme primase adds an RNA nucleotide into a primer in the 5 to 3 direction by adding one nucleotide at a time to the 3' end. DNA polymerase III then adds DNA nucleotides to the primer, making Okazaki fragment 1. After reaching the next RNA primer, DNA polymerase III detaches. Okazaki fragment 2 is then primed by primase. And DNA polymerase III again adds DNA nucleotides and detaches when it reaches the next fragment 1 primer. Next, DNA polymerase I replaces the RNA nucleotides with the DNA nucleotides by adding to the 3' end of the second fragment. DNA ligase then forms a continuous DNA strand by joining the sugar phosphate backbone of all the Okazaki fragments. This is the phosphodiester bond right here, and this new strand created is the lagging strand. And now the DNA replication cycle is complete.